Tara. Let me see if I can share my screen here. Okay. Okay, does that look right? Can everyone see okay?
Yes.
Okay, perfect. And is the volume okay?
Perfect.
Yes.
Okay, all right. So first of all, I'd like to thank Andrew and Ogechi for asking me here to speak today. I'm going to talk a little bit about the microbiome of the skin and then some defects in atopic dermatitis. So I think that the story of the microbiome really begins way back in 2007 with the NIH Human Microbiome Project. Sorry, let me just minimize this for a second. Thank you. Okay, and in phase 1, they actually looked at 5 different areas: the skin, the gut, GU tract, nasopharyngeal, and the oral cavity. And I think it was really great and lucky for us in dermatology that skin was included in this microbiome project. Of course, it is easily, the largest organ. It's the most important organ, and it's the one that's most easily accessible. But I think it was really great for us to benefit from this. There were multiple institutions involved. As you can see here, the J. Craig Ventnor Institute was the institution in San Diego that was primarily involved in this. And then in phase 2, they actually looked at disease states. They looked at diabetes, type 2, pregnancy and preterm birth, and inflammatory bowel disease. There was a lot of funding, so the institutions who received this funding were very lucky. There was about $170 million of funding for this project, and it generated a ton of data, over 14 terabytes of data. So for us in dermatology, I think that the first report from the microbiome, sort of the landmark article, was published in 2009 by Elizabeth Grice and Julie Segre, where they looked at sort of the topographical and the temporal diversity of the skin microbiome. And in that paper, they divided the entire body up into different areas. They talked about the sebaceous areas, which are kind of this light blue here on the map. They also divided it into the moist areas, like the interdigital web spaces, popliteal fossa, gluteal crease, and then the dry areas were kind of the pink ones here. So they considered sort of the volar forearm, hypothenar palms or the hands, and then the buttock. And they used DNA sequencing methods, both partial and full-length sequencing, which gave them definition to the species level. And so you can see here for the sebaceous areas, it's sort of this dark blue color, which was primarily the Proprianobacteria. The moist areas were this kind of light blue, which is Corynebacteria. And then the dry areas were the dark pink, which are the Proteobacteria. Since that time, there have been actually many sequencing advantages, and the processes that they primarily used were the amplicon method, which took advantage of the bacterial 16S ribosomal RNA. And using that, they were able to get data sort of down to the species level. Now we've had more sort of up-to-date advances, and you can use whole genome metagenomics or shotgun metagenomics, which actually enables identification all the way to the strain level, so much more specific findings. And this is one of the first studies that took advantage of those updated sort of methods. And in this study, they looked at the temporal stability of the human microbiome. And it's really an interesting study. They sampled 12 individuals across 16 sites. So they swabbed them in all these different sites and then looked at them at sort of 1 to 2 days, or 1 to 2 months, and then over a years, 1 to 2 years. And then they used this euplatin theta index to really see if the microbiome was similar to previous or if it was changing. So closer to 1 meant that it was sort of more similar to the previous finding. And this is what they found. I thought this was really fascinating. So this is the dry levels here. Oh shoot, let's see, sorry. These are the sebaceous levels here in green. This is moist, and then this is the foot sites. Here in this column is 1 to 2, I think this was 1 to 2 months. The long term was 1 to 2 years. And you can see here that at the species level, the sebaceous sites were really the most stable. Dry sites actually, surprisingly, were also stable, like the palm, which it would be sort of counterintuitive to think that, because we wash our hands and we gel our hands probably over 100 times a day, but in spite of that, the microbiome actually remains pretty stable. The sites on the foot, I guess, as you would expect if you're walking barefoot through different areas, that was the least stable site. And then the most interesting thing here also was they were actually able to take it down all the way down to the strain level. So this is looking at S. epidermidis, a specific strain, and they're able to see over the short term, it's actually quite stable here, and even pretty much so over the long term. So this kind of suggests that there is strain level stability within individuals, stating that we really maintain rather than reacquire our prevalent microbes. Other thoughts about the microbiome and changes with aging. It's thought that gut microbial communities actually stabilize around age 3, but skin microbial communities do shift, as we would expect, especially during puberty with hormonal changes. Prepubescents tend to have more streptococci, bacteroides, and proteobacteria, and then the postpubescents are going to have sort of more lipophilic microorganisms like the propionobacteria, Carinibacteria, or Malassezia. Sorry, let me just, okay. So what happens to our microbiome in common disease states? And what I'm going to do is just touch on very briefly some of the common disease states. And really, I think the story is still evolving, and a lot of what I'm going to show is very preliminary. So going back to this paper from 2009, we had noticed here in the sebaceous sites that they were primarily the Propriano species. So the first sort of disease state I'm going to talk about is acne. Propionobacteria has a new name. It's now called Cutibacterium. It was reclassified in 2016. They said because of this phylogenetic tree structure and then the DNA GC content. Cutibacterium was originally named Bacillus acnes, and then renamed Propionobacteria because it can generate propionic acid, which makes sense. It was first associated with acne in 1900 in this paper by Gilchrist, which was published in the Johns Hopkins Hospital report. And as we all know, it's a facultative anaerobe, and it survives in the anoxic sebaceous gland. But what I think is really fascinating about Cutibacterium is there are actually 6 different phylotypes. I really didn't realize this, and only phylotype 1A1 is associated with acne. And this phylotype actually carries extravirulence genes. It generates more porphyrin, generates more reactive oxygen species, and increases inflammation. Phylotype 2, which is also called P. acnes defensins, and type 3, which is the elongatum type because it kind of looks elongated when you see it under the microscope, are really just associated with healthy skin. But interestingly, when you get a deep tissue infection or a medical device infection, those are associated with the non-acne types of Cutibacterium, so type 1B, 2, and 3. And so it's thought that certain strains become pathogenic in different environments, but we really don't understand why. And so I think it's going to be fascinating to find out sort of why these organisms act in different ways in different conditions. So what about the role of C. acnes in acne? Well, they use lipases here that they produce to degrade sort of the lipid in sebum, and that causes this release of fatty acids, which then promotes adherence and sort of neutrophilic migration. They can also use proteases and hyaluronidase here to break down that extracellular matrix of the follicle, which also increases invasion and causes inflammation. They also produce the secretory CAMP factor, which upregulates IL-8, which we all know is important in neutrophilic migration. EPS that they produce acts as a diffusion barrier and actually delays the flow of antimicrobials into the follicle and also helps for comatoformation. And then the type 1 strains also carry a linear plasmid that's really essential for biofilm formation. So C. acnes, I think, is really well positioned to cause acne in the follicle, but now that we know that there are different phyllotypes, I think that the future of acne is really exciting. I mean, how long have we been giving patients Retin-A, benzoyl peroxide, and topical antibiotics? So I think that sort of this insight into more of the specific microbiome may lead to sort of more exciting treatments in the future. Some of those are, they've found that a commensal S. epidermidis actually produces succinic acid, which can inhibit C. acnes growth. So maybe we're going to be in the future putting a probiotic on our skin to prevent acne. Other ideas out there have been bacteriophage treatments, because by shotgun metagenomics, they were actually able to identify bacteriophages in healthy patients in their C. acne. So I thought that maybe we can treat ourselves with a bacteriophage and prevent acne from forming. And finally, which I think is really exciting, is this idea of a vaccine. I know that Eric Wong here at UCSD was working on this. Wouldn't it be great if you could give your child a vaccine and they wouldn't have to have terrible acne in adolescence? The targets for that are heat-killed C. acnes. Other ideas have been cell wall anchored sialidase, or CAMP factor 2 is what Dr. Wong was working on. So I think that the future of acne, like sort of psoriasis, where we had this great explosion of different treatments for psoriasis, and now atopic dermatitis we've had, I think that the future of acne is also going to be very exciting in the future. Okay, I'm going to just touch a little bit on the microbiome in psoriasis. There really is not a lot out there. This was published in 2018 in Microbiome, and they actually profiled 28 subjects with psoriasis and 26 normal subjects. And what they found was this is psoriatic lesional, non-lesional skin. These are the healthy subjects. And so initially what they found was healthy and psoriatic non-lesional skin seemed to have an increase in P. granulosum as well as P. acnes. Then they also found that psoriatic lesional and non-lesional skin in comparison to healthy subject had an increase in S. scurri, Staph aureus, and then this S. peduncophore, and that S. epidermidis here in healthy skin seemed to be increased in comparison to psoriatic lesional and non-lesional skin. This was not statistically significant, but was an interesting finding, I thought. So I honestly think that it's really unclear. You know, this is just one paper out there. There's many different papers identifying many different sort of organisms out there. But I think that the word is still out on the microbiome in psoriasis. I don't think we understand it that well at this point. I do want to point out there was a paper that was published from someone from the NIH recently. They were using an imiquad mouse model of psoriasis. They exposed the patient or the mouse to Candida first, then the imiquad, and found they had a really enhanced IL-17 response in that mouse model. So maybe in the future they're thinking Candida may play some role. So anyway, I think we really don't know the answer yet. I think it's a really interesting area for future research. What about rosacea? I think similar to psoriasis, we really don't know much about rosacea. We're so very early in the definitive story. This was a paper here, it was published in 2019. They found that there was a decrease in Roseomonas mucosa in the erythema telangiectatic type of rosacea. And then they found that pustular rosacea seemed to be enriched in Campylobacter ureolyticus and Corynebacteria and Kropinsteadi. And then this Kropinsteadi was also associated with more extensive disease. When I reviewed the literature on that Corynebacteria, all I could find was that it is a gram-positive lipophilic bacterium. It's been reported in some breast infections and in one case of endocarditis. But I think, again, we really don't know the significance of any of these findings yet. So the word is still out here on rosacea. So what about the microbiome in atopic dermatitis? I think we do know a little bit more about the microbiome in our AD patients. And I think that all the way back before most of you were born in 1974, this paper by Jim Layden and Al Kligman was really a landmark article that showed that in patients with atopic dermatitis, there is an increase in Staph aureus in these patients. And so why is Staph aureus associated with AD? Well, it's a pretty hardy organism. It can withstand a high salt content that we see in sweat. It can also utilize urea that we see in sweat as a nitrogen source, and it likes to grow in higher pH conditions. And that's sort of in contrast to a normal subject that has a lower, more acidic pH. So Staph really loves to grow in AD skin. And they also can adhere very well to the skin. They have these adhesion components of Staph aureus, which are these clumping factors, A and B, fibronectin binding protein, iron-regulated surface determinants that help them to really stick to the skin and adhere to the skin well. Staph aureus is also very pro-inflammatory. It produces protein A that induces inflammation through the TNF receptor. It has lipoproteins to induce TSLP, which, as we all know, also induces the Th2 cytokine response. It produces phenol-soluble modulins that stimulate inflammation through IL-36 and IL-17. And it produces superantigens that really trigger B cell expansion and cytokine release. It also is great at barrier destruction. It produces an alpha toxin that forms a barrel pore formation and is really critical to biofilm formation. And it also produces multiple proteases that also sort of penetrate and cause dissolution of the stratum corneum. And of course, that is going to be potentiated by the absence of filaggrin in some of the patients with atopic dermatitis. So I hope I've convinced you all then that Staph aureus is well armed against patients with atopic dermatitis. So the question then is, what is different though about our atopic patients that are making them susceptible to Staph aureus? So I think this was one of the landmark articles, was published in the New England Journal of Medicine by Rich Gallo and Donald Leung. They were able to show that there is a decreased expression of antimicrobial peptides in patients with atopic dermatitis in comparison to patients with psoriasis. So they looked at both HBD2 as well as cathelicidin, and they were able to see that there was definitely a decrease in sort of inducibility of these antimicrobial peptides in comparison to those patients with psoriasis. So it was thought that this inability to produce the antimicrobial peptides increased susceptibility to Staph aureus or other types of infections like eczema herpeticum, for instance. We took it a step further. This was a paper in 2010, and I do want to point out that our esteemed Rob Dorschner is also on this paper. We looked at patients with more severe atopic dermatitis, so those with a history of eczema herpeticum. We had 9 ADEH positive subjects. We had 21 AD subjects and 25 psoriatics. And we were able to show here that cathelicidin was statistically significantly decreased in subjects with a history of eczema herpeticum, ADEH subjects, in comparison to patients with AD. This was also true for HBD2, where we saw a decrease in patients who were ADEH positive. HBD3 almost reached significance. It was a P of 0.06, but didn't quite make it. But there was definitely a decrease in both cathelicidin and HBD2 in these patients with some more severe atopic dermatitis. So if we look then at AD versus normal skin, so the AD skin has that inhibition by their Th2 cytokines, which allows for a decreased innate immune system and thus this decrease in their antimicrobial peptide production. Their immune system is also a lot more pro-inflammatory. The pH is neutral as opposed to acidic, so that allows for better Staph aureus survival. As we all know, their barrier function is defective, which allows for sort of an enhanced entry of microbes. The Staph makes it easier for Staph aureus to stick to this disrupted barrier. And also the patients themselves scratch a lot, so they also cause a mechanical disruption in their skin. So I hope I've convinced you then that Staph aureus is associated with atopic dermatitis. So all the way back from 1974 with these culture-based methods, and now with these much fancier and much more expensive DNA-based measures, we're seeing the same types of findings. So one of the papers that sort of took advantage of sort of the fancier techniques and looked using shotgun metagenomic sequencing, looked at Staph aureus and Staph epidermidis strain diversity in patients with pediatric atopic dermatitis. And in this study, they looked at 11 children with moderate to severe AD and compared them to 7 healthy children. They sampled the patients at baseline, flare, and then post-flare. And then they performed the shotgun metagenomic sequencing. And this is kind of some of the data here. This is the objective SCORAD. So as you would expect, so baseline, which are these golden dots, are very low here with the SCORAD. The purpley dots here, you can see the SCORADs increasing during the flare, and then the teal dots are post-flare. And so when they divided it up into the different sites of the body, for instance, this is volar forearm, antecubital, etc., going across here, you can see that the purple sort of squares here really have sort of a decrease in their Shannon diversity index. You can see that during a flare, that diversity does decrease. And so here, if we look at the second panel here, you can see this is baseline, and then during a flare, it becomes very homogeneous here, sort of this light blue, and then again, post-flare becomes very heterogeneous. And they were able to show that this light blue was primarily Staph species. They sort of looked at mean relative abundance as well, and they were able to show that Staph species were statistically increased during a flare, which kind of resolved here during post-flare. They then took it a step further and they speciated it, and no surprise, during the flare, which is this dark pink, this was primarily Staph aureus. This was statistically significant when they looked at mean relative abundance. But I think one of the most exciting things actually that this paper showed was when they divided the patients between those who were more severe, who actually seemed to be Staph aureus predominant, and compared those to patients that were Staph epidermidis predominant. They actually found that the Staph aureus predominant individuals seemed to be colonized with a specific Staph aureus strain, which had never been shown before. So during flare, you can see these are the Staph aureus patients, and post-flare, it's a very sort of clonal strain here of the Staph aureus. When they looked at it for Staph epidermidis, they found that during the flares, it was kind of a very heterogeneous population. So these Staph aureus positive individuals seemed to be colonized with a single Staph aureus strain. So that was the conclusion of the paper, that clonal Staph aureus strains were seen in more severe patients, and it was more of a heterogeneous S. epidermidis strains in the less severe patients. This was a very interesting article that was published in JACI that actually looked at the skin microbiome before the development of atopic dermatitis. So it was sort of a prospective study where they swabbed 50 infants at day 2, month 2, and month 6. Then they went and they followed these patients for the development of atopic dermatitis. At the end of 1 year, they had about 10 subjects who developed AD, and so they compared those to 10 healthy subjects. And they looked at a lot of different interesting demographic data. Of course, they looked at sex. They looked at whether the patients were delivered by C-section or whether it was a vaginal delivery, how they were fed, breastfeeding, formula feeding, or if it was a combo, if they lived rurally or in the city, if they had pets, if they used emollients, how often they bathed, and if they used antibiotics. So I thought those were kind of really interesting demographic data that they collected. And this is the data here, and I thought this was really fascinating. So they found that colonization of the antecubital fossa at month 2 seemed to be, with a specific commensal staph, seemed to be associated with a decreased incidence of AD at 1 year. So this is the 2-month data that we're looking at. And these are all the unaffected individuals. These are all the individuals that went on to develop atopic dermatitis. But you can see here in the unaffected individuals, they have all this sort of dark blue here in their antecubital fossa, and these were staph species. When they went on to speciate what type of staph was present here, you can see that this was primarily all staph, a lot of it is anyway, Staph epidermidis. So there seemed to be something protective about having Staph epidermidis at 2 months, which seemed to be protective in developing atopic dermatitis at 1 year. They then went on to try to look at all the differences that I mentioned and all those demographics that they had collected. This was looking at birth methods, so C-section versus vaginal at all the different time points. These are all sort of the different locations. And unfortunately, they weren't able to see any differences in Shannon diversity by any of the demographics that they collected. But I think part of the reasons is because I think the numbers were probably too low to see any differences. But I thought it was very interesting that they looked at all of that. So I think that then you can see that the presence of these commensal staph at 2 months were protective in the development of AD. So the next question that was asked was, is there a defect in the commensal staph in these patients with atopic dermatitis in comparison to normal subjects? And this is a slide I used to show probably even 10 years ago here on the left, but now I added a little bit more here on the right about the commensal Acnes strains. But here on the left, what we were trying to show was that there's different antimicrobial peptide production in all layers of the skin. So for instance, the keratinocyte is able to produce all these antimicrobial peptides. Mast cells, Dr. DiNardo has shown eloquently, produces cathelicidin, which is one of the major antimicrobial peptides. Sebocytes produce antimicrobial peptides, neutrophils, T cells, the eccrine epithelial cells all produce antimicrobial peptides. So the skin is a great antimicrobial layer to prevent infection. But now we're learning that sort of the commensal staph way up here on the epidermis actually also can produce antimicrobial peptides. We call these the coag negative staph AM positive. So those that have the ability to produce antimicrobial peptides, because not all of them do have that ability. It's only some, and it seems to be pretty much strain specific. So the types of antimicrobial peptides that they can produce are sort of alpha helical amps, like phenyl-soluble modulins. They can produce nucleobase analog types, like 6-hydroxyamminopurine, which is firmicidin, and they can also produce lantibiotics. And I'll talk about a lantibiotic in the subsequent slide. So the Gallo lab, I think, was really ingenious in developing this high throughput screening for looking for antimicrobial activity against Staph aureus in their commensal staph. So we performed a skin swab. They then plated it on mannitol salt agar. And on the agar, the Staph aureus are yellow and the non-Staph aureus are pink. So they took all the pink colonies, grew them up in overnight in culture, and then plated them with Staph aureus in a 96 well plate, incubated that together for 22 hours, and then looked at bacterial growth by an OD600 method. And here you can see here where it's been killed pretty quickly, whereas Staph aureus still remains. So it was a great sort of quick way to determine the proportion of commensal staph on a patient that had the ability to kill Staph aureus. And I think in my 20 years or so of practice, no data has amazed me more than this graph that they showed me one day when we were having our lab meeting. And what this is this was a normal subject, and this is an atopic subject here. This is just one patient. On the y-axis is actually Staph aureus growth. And in the normal subject, you can see here, all these different clones, all of these, their commensal staph had a great ability to kill Staph aureus. So 61 out of 84 that they sampled, or about 72% of their commensal staph was able to kill Staph aureus. When they looked at the patient, this is one patient with atopic dermatitis, when they looked at their lesional skin, they actually found only 3 of these commensal staph were able to kill Staph aureus, which was about 3.6%. And then in AD non-lesional skin, it was about 20 out of 84, or 23.8%. So AD patients have such a terrible decrease in their commensal staph, in their ability to kill Staph aureus in comparison to normals. They then expanded this and looked at 49 subjects with atopic dermatitis and compared them to 30 normal subjects. And this is the data here with the total percentages now. So you can see, again, about 76% for normal subjects. AD lesional skin is way down here at about 18, and AD non-lesional skin at about 22. So the AD microbiome has a huge defect in their commensal bacteria that are able to kill Staph aureus. So the next question that was asked was then, is it possible to alter this defective microbiome in patients with atopic dermatitis? Can we correct that dysbiosis and then replace or enhance host function? So this idea that the Gallo lab had was for this autologous microbiome transplant. And what the idea actually came about was we were a little afraid that it would be hard to get approval from the FDA, because this is an IND study that we had to go to the FDA for. We were a little afraid that they wouldn't approve us to put a random bacteria on a patient. So we had the idea to take the bacteria from the patient, grow it up, and then put it back on the same patient. And the transplanted bacteria that we take from this patient had to be selected based on their capacity, of course, to inhibit Staph aureus. When Gallo first mentioned this, I thought it was a crazy idea, but I'll show you how it turned out. It actually turned out quite well. So first, what had to be done was Dr. Nakatsuji had to screen a bunch of different vehicles that would enable bacterial survival, because we had to come up with a cream that we could put the bacteria in. And that cream we wanted to be able to support bacterial survival well. So these were the different vehicles that were used. And you can see by looking at the different curves, this is live S. hominis A9, and you can see how quickly sort of it was killed in certain vehicles. But 3 of these actually supported bacterial growth quite well. And that was actually CeraVe lotion, Cetaphil lotion, and petroleum jelly actually survive, bacterial can survive quite well in it. He then put S. epidermidis as well as another commensal S. hominis in Cetaphil lotion, froze it at -80, and then thawed it out and plated it out, and was able to show that in Cetaphil lotion, S. epidermidis and S. hominis survived quite well, even with freezing and then at room temperature for up to 7 days. So Cetaphil lotion was picked as the vehicle. The intervention, so what are we going to put in to the lotion? Well, our goal was to achieve a density of about 1 × 10⁵ CFU per centimeter squared. It's kind of hard, you know, when you're in the lab and you're trying to figure out how to formulate a drug, you're not sure like what should be the right dosage for these patients. Well, it was decided that we would try to put a density that is the same as the density of bacteria that's normally found on skin. And so that was how we came up with 1 × 10⁵. So the intervention was either the lotion alone or the lotion plus about 10⁷ CFU per gram of coag-negative antimicrobial staph that was actually grown for the patient. Okay, so this is the study here, and I'm just going to kind of go through it here. And so what we did was we had to swab the non-lesional skin of the patient to try to screen for commensal staph species that had activity against Staph aureus. So Dr. Nakatsuji screened these, and then if he was able to find that they did have commensal staph with antimicrobial activity, he isolated it, he expanded it, and then put it into the Cetaphil lotion, and then we gave it back to the patient. But this was sort of personalized medicine to the nth degree. I mean, this is really amazing. So each patient had their own bacteria grown up and then put into the lotion for them. And interestingly, these were the 5 active subjects here. You can see that they all have different strains of bacteria. And this has been shown to be true, that different strains of bacteria do produce different antimicrobials. Not all of them do, but some of them do. And you can see here too, this is a different, concentration of their bacteria. And you can see they all have sort of different killing rates there. So a very personalized medicine that Dr. Nakatsuji was able to make here. And the other thing I wanted to mention is he tested this against each patient's own Staph aureus. It wasn't against the stock strain of Staph aureus. It was actually each patient's own Staph aureus. And then this was the trial here. We had 14 patients that were enrolled. 3 actually had to be screen failed because they didn't produce any CONS that were antimicrobial. So they unfortunately had to be dropped out of the study. So that's how we ended up with 5 in the active arm and 6 in vehicle. So what we did was on day 0, we swabbed them for baseline. They came in mid-treatment at day 4 to get more drug and to get repeat swabs. Then they came in again at day 7, and that was the end of treatment. So they did not apply any more medication after day 7, but they continued to come in on day 8, 9, and then day 11. And we swabbed them at those times. So we were really excited to see that in lesional compared to non-lesional skin, we saw this nice statistically significant decrease between vehicle and active in lesional Staph aureus. This y-axis here is sort of relative Staph aureus CFU, and you can see it suppresses Staph aureus quite well. It's significant at day 3 to 4 mid-treatment, end of treatment, and even off treatment for both lesional and non-lesional skin. And I was also very surprised to see that I didn't think that we had enough time in this trial to see improvement in local EZ, but you can see here at day 3, 4, you start to see sort of a widening between vehicle and active. Day 7, you can see even more improvement in the active arm, and then it reached statistical significance off treatment, so days 8 to 13. So we were really excited about that ability to decrease Staph aureus as well as improve the local EZ. And so because of that, we decided to try a much larger trial. And this is what we call the targeted microbiome transplant. And this enrollment goal was actually pretty high. It was 54 participants, and we decided to enroll them in a 2-to-1, so 36 were in the active arm and 18 were in placebo. But because this was such a large trial now, there was no way we were going to be able to grow up each patient's own bacteria and formulate it in a cream. So we had to figure out a specific bacteria that we felt would be the most safe and the most effective for treatment of these patients. We did have to screen 82 subjects to randomize 54 because a lot of the patients were not Staph aureus positive. They had to be Staph aureus positive to be in the study. So that's why we had to screen so many patients. And the NIH really wanted the primary endpoint to be safety because this was a phase 1 trial. So our primary endpoint was sort of adverse events. The bacteria that was chosen, these are all different healthy donors here. The positive means that these were the ones that had activity against Staph aureus. And it was this healthy donors, S. hominis A9 that was chosen to be put into the lotion. Looking at the antimicrobial peptides that were produced by S. hominis A9 by HPLC, they fractionated at 30 and 32 and were found to be a 3.1 and a 3.5 kilodalton fragment. Now, Dr. Nakatsuji and Dr. Gallo decided to name this antimicrobial peptide Hogocidin. And I was really confused why they would name it Hogocidin because as antimicrobial, a hog really didn't come up in my mind as being antimicrobial. But it turns out that in Japanese, which Dr. Nakatsuji is from Japan, that translates to protector in Japanese, and thus the name Hogocidin for this antimicrobial peptide. This was just showing a nice inverse correlation between relative Hogocidin and Staph aureus growth, so it is very effective against Staph aureus. And this was the study design. I know I'm running out of time here, so I'm going to try to go a little bit faster of this trial. This was the screening phase, very similar to our previous. We did perform the baseline swabs. Everything was the same. We did mid-treatment swabs at day 4, and then again at day 7, and then the clinic follow-up visits at day 8, 9, and 11. These were the demographics of the patients. I was going to talk a little bit about dosage calculations, but I'm going to skip over that just because in the interest of time. The primary endpoint was the count of serious and non-serious treatment adverse events between day 0 and day 8. And I am going to just show this data here. The per participant event rate for the TMT, which was the active drug, was 0.19. For placebo was 0.34. So there was no statistically a significant increase in any adverse event in comparison to placebo. If you look at this though, we did have kind of a high adverse event rate. TMT was 63 and placebo was 55. And I'm going to go just a little bit into why that was. This is looking at sort of treatment adverse event summary. So the good news was there were no serious adverse events. There were no deaths. There were no adverse events that led to treatment discontinuation. We did have one moderate event in the TMT arm, which was just a furuncle that developed on the gentleman's arm 24 hours after the end of the study, so it was day 8. It resolved without antibiotics with just warm compresses. Placebo had 2 moderate adverse events which were like a GI upset and a hordeolum. But again, we did have 61% in TMT and 83 in the placebo that did have sort of adverse, mild adverse events. And the reason for this was because we had them fill out a daily diary every day where they reported fever, chills, or night sweats, which was recorded as an AE. But I think that the reason that we had so many was primarily because they also, in their right and their left arm, had to report pain, swelling, or eczema every single day. And so if they had if they marked it a 5 one day and they marked it 6 the next day, that was considered an adverse event. So we sort of captured the normal variation, I think, that we do see in patients with atopic dermatitis. And I think that's why the rate was so high. But luckily, the TMT active arm was actually lower than the rate that we saw in placebo. This is again just showing it by organ class and preferred term. So you can see that the eczema, atopic dermatitis, pain, and then swelling kind of was the reason why all these numbers were so high. Again, though, these were not statistically significant. This was close to significance, but it was the opposite way. There was kind of a decrease in the number of adverse events in TMT versus placebo. Again, this is just more of the adverse events by organ class. Again, you can see that, again, all the numbers are very low. There was really no signal here. The p-values were non-significant. And then this was the great news that TMT did seem to suppress Staph aureus. This is the relative change in Staph aureus. This was time over here. So you can see mid-treatment, again, there was a statistically significant decrease in Staph aureus in comparison to vehicle. And this was significant all the way through for lesional skin and almost all the way through for non-lesional skin. I think at day 8 kind of did not reach significance there. And then looking at local EZ, we were able to show that there was this percent change in local EZ from baseline. So here, this was placebo, this was active drug, and you can see that there was a statistically significant improvement in local EZ day 7, day 24, as well as day 48. And then this was beautiful. We were able to show that there was a change in local EZ from baseline that correlated very well to change in Staph aureus. It was very significant. This is for lesional skin, and this was not, I'm sorry, this was for active drug, which was not shown, of course, for vehicle. So we concluded then that TMT is really safe in patients with AD. It does suppress Staph aureus survival, and application of the TMT also improves the local EZ score and correlated very well with the decrease in Staph aureus. We do have a lot of future directions. We have a commercial preparation of S. hominis A9 that has a greater than 1 year shelf life at room temperature, so it'll be much better than shipping things at -80, then thawing them out, and then having the patients come back mid-treatment to get more drug. We can just give the patients the drug because we do have a long-term clinical trial planned. It's going to be a 3-month trial, and we want to see if maybe putting this drug on for a longer time can possibly have a longer lasting effect on the microbiome. It would be amazing to see if we could actually change, for permanently their microbiome, but we'll have to see if that's going to be true. We also have a short-term clinical trial plan to look to better assess sort of the kinetics of S. hominis A9, and also look at it in Staph aureus negative in normal subjects, because we don't know if that's going to have allow the transplant to survive longer or not. And then we're also going to look at the effect of preconditioning. Maybe if we precondition these patients with mupirocin and we kill off all the bacteria there, maybe the transplant will last longer, or possibly preconditioning and reducing inflammation with triamcinolone may also make the transplant last longer. Finally, with Dr. Hightower and Dr. Eichenfeld, we plan to look at this in the pediatric population, to look at sort of what age this dysbiosis of the microbiome begins. I think it'll be really interesting to see when they start to have that shift in their commensal staff. And then we're also going to have a trial applying this to the pediatric subjects, primarily to see if it's safe, but also to see if it can improve their clinical AD. I do want to thank, of course, Rich Gallo and Taro Nakatsuji for making all of this possible. I also want to thank Faiza Shafiq, my lead coordinator and really backbone of our clinical trials unit. My current fellow, Joyce Chang, as well as Larry Tong and Kim Chun, all of which worked tirelessly on this trial, coming in on weekends to collect that day 9, day 10, and 11, getting locked in the garage because the elevator doesn't work on the weekends. Just crazy things happen to us. And I do want to thank Anna Butcher, Paul Kodal, and Carlos Aguilara, who are also the key collaborators in the lab, as well as Donald Leung, who is the PI of the ADRN Consortium, and Patricia Taylor, both at National Jewish. And then I was going to close with this wonderful pictures of this hike that we did, but I'll just scroll through them quickly. I know we're out of time. I can definitely take questions from people maybe offline. But what I was going to say was, I think research is a lot like hiking. It's a very hard and arduous process, but it's a lot of fun along the way, and it's great when you get the results at the end. So I thank you all for your attention.
Thank you, Dr. Hata. That's really impressive and exciting work. There are a couple questions in the chat. Did you want to respond to them or reply back in the chat?
Yeah, maybe I will, because I know we're out of time. I will, let's see. Why do you hypothesize you have to apply and the improvement does not persist? We've actually found that in lesional skin, the inflammation in the skin seems to kill Staph hominis. And so that's why we're trying to see if maybe decreasing that inflammation will promote longer longevity of the probiotic. So that's Dr. Scheinberg's question here. He's asking, why do we hypothesize that you have to apply the probiotic twice daily and the improvement does not persist when you stop the treatment? What's the difference between atopic and normal skin? And I think that primarily the difference is that inflammation in the atopic skin that's basically killing off S. hominis A9. We found that S. hominis A9 actually is not as hardy as some of the other bacteria. So we're looking at ways to sort of improve longevity in that way. Comment on adding vaginal flora to skin. Yeah, I am not sure about that. I think that's a really interesting question, and that's why I was hoping some of that data, that's why I put that one slide in there about vaginal delivery versus C-section, but I actually think that I had tried to look into that because I was going to try to add a slide about that into this talk, and I could not find any differences in improvement in AD anyway with the patients who were delivered vaginally versus C-section. So, okay, I think that's it. Should I just end here, Tara, or?
Any other questions from the group that anybody, I know we're, okay. No, thank you.
I had this great inspirational talk I was going to give with those slides at the end. I'll give that another time.
We would love that. We would
Okay.
love that. Thank you.
All right, guys. Thank you all so much. Appreciate
Bye-bye.
it. Goodbye.
Thank you, everyone.